So today our topic is cell wall. Cell wall forms a fairly rigid layer outside the plasma membrane in most of the prokaryotes. The cell wall accounts for 10 to 40% of the cellular dry mass depending on the growth of conditions. It provides the cell support and protection against mechanical stress or damage from osmotic rupture and lysis. The major component of the cell wall, bacterial cell wall, is peptidoglycan or murin. This structure, this is a structure of a peptidoglycan specific only to prokaryotes and gives the cell shape and surrounds the cytoplasmic membrane. Functions of bacterial cell wall. The bacterial cell wall performs several functions as well. In addition to providing overall strength to the cell, it also helps maintain the cell shape, is important for how the cell will grow, reproduce, obtain nutrients, and move. It protects the cell from osmotic lysis as the cell moves from one environment to another or transports its nutrients from its surroundings. Since water can freely move across both the cell membrane and the cell wall, the cell is at risk for an osmotic imbalance, which could put pressure on the relatively weak plasma membrane. Studies have actually shown that the internal pressure of a cell is similar to the pressure found inside a fully inflated car tire. That is a lot of pressure for the plasma membrane to withstand. The cell wall can keep out certain molecules such as toxins, particularly for gram-negative bacteria. And lastly, bacterial cell wall can contribute to the pathogenicity or disease. A space is observed between plasma membrane and outer membrane of cell wall. It is called periplasm. Periplasm gram-negative bacteria contains hydrolytic enzymes. They facilitate the movement of molecules across the membrane. The gram-positive bacterial cell wall contains peptidoglycan and teichoic acid as the major constituents, whereas that of gram-negative bacteria possesses peptidoglycan, lipopolysaccharide, lipoprotein, and phospholipid. Cell wall of gram-positive versus gram-negative bacteria. Gram-positive bacteria, teichoic acid, surface protein, teichoic acid. Linked to another bitetrapeptide that extends off the NAM sugar unit, allowing a lattice-like structure to form. The four amino acids that compose the tetrapeptide are L-alanine, D-glutamine, L-lysine, or mesodiaminoaminopimulic acid, and D-alanine. In many gram-positive bacteria, there is a cross bridge of five amino acids such as glycine that serves to connect one tetrapeptide to another. In either case, the cross-linking serves to increase the strength of the overall structure with more strength derived from complete cross-linking. With every tetrapeptide is bound in some way to a tetrapeptide on another NAG/NAM chain. So we'll repeat the topic again. Cell wall. Firstly, we talked about cell wall. Cell wall forms a fairly rigid structure just outside the plasma membrane in most of the prokaryotes. Cell wall accounts for 10 to 40% of the cellular dry mass depending on the growth conditions. It provides the cell support and protection against mechanical stress or damage from osmotic rupture and lysis. The major component of the bacterial cell wall is peptidoglycan or mucin. This is a structure of peptidoglycan specific only to prokaryotes, gives the cell shape and surrounds the cytoplasmic membrane. Functions of bacterial cell wall. The bacterial cell wall performs several functions as well. In addition to providing overall strength to the cell, it also helps maintain the cell shape, which is important for how the cell will grow, reproduce, obtain nutrients, and move. It protects the cell from osmotic lysis as the cell moves from one environment to another or transports in nutrients from its surroundings.